Hello, my name is Brandon Coffey, and today I'll be lecturing on the identification of mycobacterium. The goal for this lecture is after this lesson, students will be able to identify mycobacterium colonies on media and identify the genus Mycobacterium with a Kenyan acid-fast stain. Upon completion of this lesson, the student will be able to list a minimum of 4 safety precautions that should be taken when handling mycobacteria samples in a clinical laboratory. Select colonies growing on media that, based on their morphology, are likely to be identified as Mycobacterium genus with 70% accuracy. Given the proper equipment and media, demonstrate the steps for performing a Kenyan acid-fast stain without missing any steps. Given a microscope, identify whether the isolate is acid-fast positive based on color and arrangement with 100% accuracy. And list 2 examples of other genera of bacteria that could result in an acid-fast positive state. So here I have some pre-test questions for you guys. Question 1, which of the following media is selective for recovery of Mycobacterium species? And the answer to that is B, 7H11 agar. Question 2, which of the following is a stain that identifies Mycobacterium? And the answer to that one is A, the Kenyan acid-fast stain. And question 3, which of the following describes the microscopic morphology of a Mycobacterium stained with a Kenyan acid-fast stain? And the answer to that one is also A, pink or red banded or beaded rods. Some general characteristics of the genus Mycobacterium include that they are strict aerobes. The pathogenic species tend to grow more slowly. Some examples of that are Mycobacterium tuberculosis and Mycobacterium avium complex. The rapid growing species tend to be less pathogenic or only sometimes pathogenic. An example of that would be Mycobacteria fortuitum, which can sometimes cause skin and wound infections. When acid-fast stained with Kenyan or Zeal-Nielsen method, Mycobacterium produces characteristic red-pink colored rods with banding, beading, or pleomorphic morphologies. Mycobacterium is a biosafety level 3 organism, which means that it can potentially cause fatal human illness and can be passed by aerosols. So there are some special safety precautions that need to be taken when handling mycobacteria in the laboratory. So mycobacteria laboratory room should be separated from the rest of the main lab and have its own ventilation. The room should be negative pressure, meaning that air is able to come in, but it's not able to leave the room. Mycobacteria samples should be handled under a laminar flow biological safety cabinet. It's a level 2 biological safety cabinet. And universal precaution should be maintained. You should wear your mask and face shield, gloves, things like that. Mycobacteria species fall into several different categories based on their photoreactive qualities. So the 3 categories that they can fall into are non-photochromogen, photochromogen, or scotochromogen. So non-photochromogens are absent of pigment regardless of their exposure to light. Photochromogens, pigment production is dependent on exposure to light. So if those organisms are kept in the dark, they will remain a buff color. If they're allowed to grow in the light, then they will produce pigment, yellow or orange pigment. Scotochromogens, they will produce pigment regardless of their exposure to light. So generally, those organisms are always going to grow a yellow or orange color. So there are some examples of the colony morphologies here. So generally, mycobacterium is going to have like dry, rough, irregular, waxy, wrinkled looking colonies. They're these kind of odd looking, almost fungal like looking colonies. And then you can see some different examples of the different chromogenic properties here. So Mycobacterium tuberculosis is a non-photochromogen. Kanzasi is a photochromogen, and Scrofulacum is a scotochromogen. The Kenyan acid fast stain. Mycobacterium can be identified by its ability to retain carbofuscin primary stain when decolorized with acid alcohol. So when you're doing a Kenyan acid fast stain, you apply the carbofuscin, and mycobacteria is able to retain that stain when the acid alcohol is added. So here's a list of the procedures for the Kenyan acid fast stain. I'll go through them quickly, and then I have a video that I can show you guys. So you're going to flood the slide with the carbofuscin and allow it to stand for 5 minutes at room temperature, and then rinse that with deionized water. You'll then decolorize with acid alcohol for about 3 minutes. And the way you're going to do this is you can add it to the slide and then remove it, and then just keep adding it until for about 3 minutes until you see that it's no longer releasing color. After that, you'll rinse it again with deionized water, and then flood the slide with methylene blue counterstain for about 1 minute. Then you'll rinse it with the deionized water again, and you'll be able to view it under the microscope after.
Before beginning the acid fast stain, hard heat fix your smear. Begin the acid fast stain by flooding the slide with carbofuscin. Let it sit for 5 minutes. Rinse the slide with deionized water and shake off the excess water. Decolorize the smear by flooding the slide with acid alcohol. Flood the slide with acid alcohol, wait 30 seconds, tilt, repeat this procedure for a total of 2 minutes. Rinse the slide with deionized water, shake the excess water from the slide. Flood the slide with 0.3% methylene blue. Let it sit for 1 minute. Rinse the slide with deionized water. Shake off the excess. Now blot. Now you're ready to view the slide underneath the microscope.
And here's some examples of the results that you can see with a acid fast stain. So you can see here, this is the morphology that you're looking for a mycobacterium species. You have these kind of long, slender rods, usually with some beading. These ones have characteristic bands. This one's actually Mycobacterium kanzasi. Some other species can have like an acid fast reaction. And you can see that it kind of has a little bit of blue and a little bit of pink in it. So this is a partially acid fast positive reaction. And then this is just a negative reaction. This is a Bacillus subtilis. So here's other acid fast positive species. So I talked about the nocardia already. Rhodococcus can be partially acid fast positive as well. And you can also use modifications of the acid fast stain to get staining on Cryptosporidium species and on endospores for Bacillus species. And to finish up, I have some post-test questions for you guys. Question number 1. Describe the colony morphology of the colony seen here. these colonies fit the description of a mycobacterium species? And the answer is the colonies are dry and rough with irregular edges and buff color, and these colonies do fit the description of Mycobacterium tuberculosis. Question 2. Explain why the Mycobacteria lab is placed in a separate room from the rest of the lab with its own ventilation, and why samples must be processed under a laminar flow cabinet. And the answer to that one is Mycobacteria is categorized as a biosafety level 3 organism, meaning that it may cause serious or potentially fatal disease in humans and can be spread through aerosols. Containing mycobacteria to a laboratory with its own ventilation and using a laminar flow hood reduces the risk of aerosol spread to the laboratorian and the environment. And the last question is, describe the following acid-fast stain results. Are they consistent with the genus Mycobacterium? And the answer is they are reddish pink rods with cross-banding. These acid-fast results are consistent with the Mycobacteria genera, specifically Mycobacterium kansasi. And here's my references. All right, everybody have a good day. Thank you.